Hi, welcome again to the course of structural biology. We are still going through the introduction module, and today is the last lecture of this module. Today we will talk about post genomic era, its effect. We have already talked about how gene sequencing made us convinced and revolutionized the field of biology. We start reading at least one macromolecule, the DNA, and then it leads to the great revolution of biology, the Human Genome Project. So, how the advancement happens? First, as I told, reading macromolecular sequences was one of the biggest dream to the scientists to achieve with numerous challenges. For a long time, we wanted to read these polymers through the reading of the monomers, but this was possible. Several legendary work was discussed in our previous lectures, which have zeroed down to DNA as the readable molecule. We have shown stage wise, history wise, why DNA has become the most essential molecule in the world of biology. When it was zeroing to DNA, it was speeded up with advances like electrophoresis technique in time of DNA sequencing. At the beginning, electrophoresis technique separation was a challenge, which was then become advanced, become capillary, become automated with laser technology. Fluorescence spectroscopy as well as labeling technology become advanced. PCR invented by Carey Mullins is one of the greatest advancement towards this, and last but not the least, the DNA sequencing technique by Sanger, Maxim, Gilbert. So, that lead to Human Genome Project. About 30 years ago, the researchers in 1980s around, and like it started at 1990, other business, industrial as well as political stakeholders started setting up the first genomic initiative, the Human Genome Project. What was conceived as an audacious plan in the 80s turned into an official multi-center international program in 1990 and was brought to a conclusion in 2003. Though we discussed, but I still want to summarize what was the outcome of this Human Genome Project. The number, location, size and sequence of human genes is now established. We know that only 2% of the human genome contains genes, the part of DNA that encode recipes of the protein, and rest of that we know junk. Interestingly, we don't know what are the remainder does. So, we know that 2% is giving us protein. Interestingly, with such a long history of research in the field of DNA, in the field of genome, we still only working on 2%. Isn't it very interesting? So, that we came to know. We have an estimated 30 to 40,000 genes. The functions remains unknown for many of them. And almost half of all proteins predicted from human genes share similarities with fruit flies and worms, which tells us how the life is a united thing, is a evoluted thing. And then after Human Genome Project, they are arriving the post genomic era. Definitely, I am not an expert to go into details, but I am just touching those things so that you could follow the how biology is proceeding. So, what is post genomic era? When the Human Genome Project was launched, it was approximated much longer time than what it actually required, because there are many technical like limitations, restrictions that time. So, people estimated much longer time. This is due to extensive technological improvement in the sequencing and further processing technique that it finished within 13 years. And as I told, these the advancements, though we say it's because of Human Genome Project, what we mean by that is when Human Genome Project was happening, it was a perfect combo of people from basic science background and people from technological background coming together, working together, thinking together, sharing their thoughts, and that leads to a lot of improvements, lot of new innovation, that and as a result we have elevated ourselves from the genomic era to post genomic era. The post genomic era refers to the time period after the completion of Human Genome Project to even today. So, in the post genomic era, the research involves applying genomic tools and knowledge to more general problems. So, we solve the genomes, but from genomes we want to go further, we want to solve meaningful problems, asking new questions tractable only to genomic and post genomic analysis. So, let us say we have done the genome sequencing, we have seen some abnormality, but how we know this is an abnormality? We do not have a standard. Now, when we have so like many standards coming, we are doing something called comparative genomics. That is opening a huge area. Why? Because now we know this is the genome of a normal person or a normal organism. So, if we see some changes, even if we do not know or do not understand, we know that our focus should be on this gene because there is some changes happening. So, that identification, that focusing is one of the greatest success and one of the greatest initiative in the post genomic era, and pushing our journey from sequence accumulation, like getting a lot of sequence, to understand what those sequence or those changes in the sequence are giving us functional inputs, how it relates to functionality. Now we realize that any changes are happening, there are some changes in the genetic level, but how to understand them is the major target of post genomic era. This is the slide you know, but I am coming to that because this is what post genomic era talked about. We already know that DNA gives DNA through replication, DNA gives mRNA through transcription, and mRNA gives protein through translation. But the following up from protein to sugar, nucleotides, amino acids, lipids, the small molecules which are metabolites, which gives us actual track to the function. So, the genome we get from sequencing, what is called genome sequencing, then transcriptome sequencing, and transcriptome gives us the proteome, and then we do metabolic studies now through newer technologies like mass spectrometry, like high resolution NMR. I will talk about them little bit though, but to just to get to understanding the link between how the metabolomic research is connecting the journey of genomic research. So, as I told, our journey is from DNA sequencing, and when we get the DNA sequence, we get the protein sequence, and we want to achieve function. So, the greatest barrier here is to get the structure. So, our goal is to understand the function. Towards getting the function, we need to get the structures, and that is the importance of structural biology. So, the challenges, how to get 3D structures, I was continuously talking about, and we reach kind of in that situation. I also want to talk about a different problem which have given a new field to explore, to prosper in this post genomic era. How could we save and communicate with those data? So, now in the era before the genomic sequencing happened, biological studies were very challenging. You were studying biology, so it have challenges of how the molecules are stable or not, how you could reproduce the situation in laboratory condition, how the even the mood of the organisms you are studying are there, how you collect things, all those things were like coming as a challenge. But now after genome sequencing is becoming easier day by day, affordable day by day, data is coming in a huge way, So that actually leads to generation of a field where this data have to be kept in a very, very proper way. Also, there are different type of data. I will discuss about them, but if you think, you will understand genome sequencing is DNA, ATGC, same as transcriptome, but there is difference in genome and transcriptome, right? Then you come to protein, which is totally different, 20 amino acids. Then you come to structure, which is coordinates. So there are different type of files. You have to shape them differently. You have to make them interact between each other. You have to extract data from them, and that huge data give birth to a field which as a whole we call system biology. Yes, this is not a place where I am discussing system biology, but I am talking about the juncture from where I am proceeding towards structural biology, and another journey started which goes towards the management of data and proper communication between the data. This is a very, very critical thing to understand. So what happened, what God is thinking now, because we told after the genome was sequenced, the code was decoded, God was really worried. So after genome sequencing project, now we have millions and billions of gene sequence, which means we have that many protein sequence known, but we need to know the three-dimensional structures of those protein, how they interact properly, how the protein interact with other protein, how the protein interact with other macromolecules like DNA, RNA, how the protein interact with small molecules like substrates, inhibitors. So that makes God happy, because one genome, many protein, and also many protein behave differently in different situation. So now he is not that much worried, rather he is saying perhaps I do not need to change the password even now, because humans still have a dynamic proteome code to break. They cannot hit a moving target. So who are the biological macromolecules which could be studied and explored? Are DNA, RNA and protein. All are important, but protein is the most important macromolecule. Why? Because if you see, proteins are functional. Protein actually talks about what or how we walk, how we stay, how the diseases are coming, and more importantly, protein function could only be understood through their 3D structure. And if you look, this is a good example where I have given different proteins. All the different protein have different 3D structures, unique 3D structures. Whereas when we talk about DNA and RNA, they do not have such diversity in their structures. So that and many other factors, which are the topic of our next module's discussion, will show us very clearly why protein structure could be the major focus of structural biology. So what are the challenges we are going to handle? What is the importance of folded protein structure? How we could determine the protein 3D structure, and why it have taken so much time and money to solve 3D structure of protein. So if you look at, it's a old graph we have already talked about. In around 2015, the journey of protein structure was surpassed by the genome or protein sequence determination, and then it rise and now there is a huge difference, and every day and night the difference is enhancing. So we also want to understand why that much difference is happening between the determination of protein structure versus determination of protein sequence. Now because we understood the basics of genome sequencing and transcriptome sequencing, we know the reason why it is becoming rapid, why it is becoming like easier day by day, but we will take a look about the structural biology technique in a very basics in this module, and why the huge amount of money and huge amount of time there. But before going to the topic, I will also want to show you about the data science I talked about. Here is a quote from Albert Branscombe. He told, "You must recognize that someday you may need as many computer scientists as biologists in your biology lab." That is what the importance. And Craig Venter, whom you probably remember, because when we talked about Human Genome Project, he have taken a private initiative of Human Genome Project. So at TIGR, we already have twice as many computer scientists in comparison to people who are expert in the biology research. So biology research is now trending into another direction, the data science, the computer science. Coming back to our question, why so much money and time required for protein structure? So first we have to know about the techniques which are used for determining high resolution 3D structures. They are X-ray crystallography, NMR and cryo-electron microscopy. So what are the common steps in structural biology? First, you need to do cloning. So that is where the first differentiation starts. When you need to do gene sequencing or genome sequencing, you directly isolate gene or genome and go for sequencing. Whereas when you want to work with protein, first you have to produce your protein in a exclusive way. I will discuss in detail, but the first part is, for doing protein you have to go back to DNA and you have to do a process called cloning, by which you could produce your protein in a high way. So cloning is a technique which help you to put your gene with some situations which will help you to control the further proceeding, the overexpression and the purification. These are mostly critical and these are the differentiable factors which takes a lot of time. As I told, after you do the cloning, it comes expression. So as I generally say, if you think you want to express a protein, but a protein is, if you want to grow the organism, if it have 5,000 protein, so your protein is, the chance of producing is 1 by 5,000. This is a critical factor, if you have to understand that. So cloning is the technology which is pushing towards the expression of your protein. And then you need purification. When you are dealing with DNA, as I told, DNA is made of ATGC. So the combination permutation in the DNA helps, and with the fact that DNA have phosphates at the surface, helps you to purify DNA, and also you could run through electrophoresis gels and take it out. So purification of DNA is much easier, whereas purification of protein, the major thing is a molecule linked by covalent bonds, right? Not here the only factor is covalent bond. In protein you have to give the importance of non-covalent bonds like hydrogen bonds, salt bridges, hydrophobics, like stacking, metal interactions, we will talk about those. So, to maintain those, you have to take real measures. So, that is the first step even before you enter in the determination technique. Cloning, expression and purification would make a significant difference in time and cost. If you ask me, for a novel protein, for a new protein to start, you have to do the initial optimization, engineering, then cloning, then expression and purification. I would take, in a normal small laboratory like ours, individual laboratories, if it is not a high throughput process, it would at least take minimum of 1 year, but you cannot predict how long it could take. So, that is one of the biggest challenge towards the journey of protein sequence to protein structure, is to prepare the protein in a purified way. Now, if you look at the steps in protein X-ray crystallography, you get a pure protein, you see the band, the pure single band will determine it is a pure protein. As a crystallographer, I will talk you something else also, which I will again go into detail when I am talking about the detail of the technique. When you are doing crystallography, even if you have a purified protein and you store it in the refrigerator, when you want to set up the drops, you should perform a size exclusion chromatography. Why? Because when you are doing the freezing and thawing, there is a high probability that protein might have at least even slight partial denaturation, and this could be deadly towards development of crystallization. If you run a size exclusion chromatography, it would not only purify your protein, it would ensure that protein are properly folded. So, one of the must thing is to go through a size exclusion chromatography just before you are setting crystals. Then you are going to set the crystals. There are many techniques I will discuss, but with all the techniques, with everything being standardized, even the world's best crystallographer could not tell you what is the condition of crystallization. This is one very uncertain step in the success of protein crystallography. You will see that we even do not consider crystallization as a science, because it is on luck, we do other things perfectly, and then in the name of God we just keep setting more and more conditions. I will discuss it in detail. So, crystallization is one of the most critical step in protein crystallography success. Then you get crystals. Are you done? No. You have to keep checking the crystals, they are diffractable or not. Then the crystals are diffracted, you will get the diffraction pattern. Then you have the many problems, but one of the significant problem is phase problem in crystallography. Again, we will discuss it in detail. If you pass through that, you will get electron density, then you will go to build the model, and finally you would get the refined model. So, many steps, many challenges. Coming to the second method, nuclear magnetic resonance. In nuclear magnetic resonance, you need a labeling with the odd numbers, like H1, C13, N15, P31, and it detects and assigns the chemical shifts of atomic nuclei with non-zero spin. So, because of the odd number you get chemical shift, and from there you detect. The shift also depend on their electronic environment identities and distances of nearby atom. I will go in detail, but just to say that suppose you have a hydrogen and another hydrogen. So, these three hydrogen are in the same chemical environment. But this hydrogen is different. So, you get different spectra, the shifting of a spectra that is defining the chemical environment. The system could be tuned to look at specific feature at the characteristic spin moment, and then you could also get the coupling where you have two hydrogen in a certain distance, you get nuclear overhauser effects. We will discuss about all those basics, how the spectras are developed, and why I am talking about hydrogen or proton, because it gives you the natural like non-zero spin. So, it is present in protein, which is our target, or any even small molecules, polymers, anywhere. So, for primary detection, or we say that one dimensional NMR, we use proton. I will talk about the technological details, but let us come to the what happen in protein. Better resolution is obtained when the molecule is tumbling fast. So, the resolution depends on the movement, more sharper the movement, higher the resolution. For small molecule it is not a problem because they are small. So, they in the solution they tumble fast. But when it comes to protein, here the problem starts. First of all, it must be soluble at very high concentration, and it could not be aggregate, and after that there is a restriction of molecular weight. So, like with increasing molecular weight, the tumbling would be slower, hence the spectra start overlapping. So, NMR have a problem of going into higher molecular weight protein structure determination, but it have certain advantages also, and we will discuss that later. But let us come to the steps. I talked about protein purification. Protein purification gets even more complicated because here you have to use the isotopes. What is the isotopes? Remember I talked about we need those. So, this is natural, but they are not natural. C12 is natural, N14 is natural, and P32 is natural. So, you have to grow them in presence of those isotopes, and that makes two very like certain things. One, it would be very costly because you have to grow them on a solution filled with those isotopes, and growing with those isotopes the protein production might be lessen, reduced, or even protein could not at all produce. So, those are the extra challenges. Again, you need the purified protein in very high concentration. Then you come to data collection with NMR. You will collect different dimension data, 1D data, 2D data, 3D data. I will try to explain everything. So, different dimension data collection. Then coming to data processing, and finally the model. Okay. Coming to cryo-electron microscopy, cryo-electron microscopy is a type of transmission electron microscopy that allows for the specimen of interest to be viewed at cryogenic temperature. X-ray is a diffraction, NMR is a spectroscopy, cryo-electron is the microscopy. Three techniques, three different ways, all contributing to the structural biology. Cryo-electron started with really low resolution, but following years of improvement, the cryo-electron microscopy has become a very valuable tool for viewing and studying the structures of various biological macromolecules, and especially it draws current attention due to recent developments. Now people are getting structures at extreme high resolution. In addition, the recent Nobel Prize have create huge interest in the field of cryo-electron microscopy. For cryo-electron microscopy, one of the biggest problem was the machine was very costly. So very few institutes in the world have developed cryo-electron microscopy facility. But the recent Nobel Prize, which I will talk in details when I will go to the technical details of the technique, these have changed the scenario. Every country now initiating cryo-electron microscopy, people start understanding its potential, and in India currently there are 8 institutes who have cryo-electron microscopy facility. Again, I will talk about the institutes and all when I will be in the technical details. So, while cryo-electron microscopy encompasses a number of experimental methods, it could image intact tissue sections, imaging plunge frozen cells, image intact viruses, and image macromolecular assemblies like ribosome. So, as I told, when we are talking about protein crystallography, it is very efficient, provide very high resolution crystallography from its starting time, but somehow it is restricted to the proteins. NMR have definitely other applications, but it have restriction of size. And especially if you see, when our journey of viewing is going on the next level, like we have learned about the pieces, now we are more interested to assemble the pieces. We want to look at the holistic picture. Cryo-electron microscopy is definitely an instrument, a technique of future. I will talk about details. Steps: sample preparation, then the sample will be going to grid preparation, the grids it would be frozen, and then the grids will be coming under the microscope, and the thinner sections would be collected, image would be collected as a 2D image, and then the different 2D images would be matched, aligned and averaged, which will give a 3D map, and that 3D map would be refined to 3D model. So, I talked about these 3 techniques. Now come about the time and money. First of all, as I told, one of the main problem is when you are working on DNA, and from isolation, purification, much easier. Whereas when you are working in protein, you have to go back to DNA. You have to work on the DNA so that it could express, because for DNA, if you get a very tiny amount, you do not care, because you have carry Mullin's PCR, which will enhance the amount of DNA as much as you want. Here you have to use biological systems which will help you to grow. So, what you will use? Obviously, the first choice would be prokaryotic system like E. coli, but sometimes there are proteins which could not be expressed or could not show the required post translational effects, modifications when you are working with those proteins. You have to go to eukaryotic systems. Your challenge would be even more with the increasing price of the media, maintenance of the cells and all these things. So, the cloning, overexpression, purification is a big problem. Also, when you are dealing with eukaryotic cells, you have to continuously reproduce them. So, you cannot stop your lab in any natural calamity, in any reason. You have to keep reproducing if you want to continue them. That is another challenge. Then, so this is a challenge for all the instruments. Then talking about crystallography. The first challenge, as I told, is crystallization. Nobody understands how long it would take. Nobody knows which condition would be favored. A lot of research happened on them, a lot of statistical data people have explored, people have developed screening set. Definitely Hampton have the monopoly, but currently there are new companies are coming with their research oriented kits, but at the end the scenario is kind of same. You have to keep trying. So, crystallization is one of the major challenge, but then there is also phase problem, also problem of tiny crystals. All of them keep things time consuming, and all of them are bringing more money to the system. Then you have good diffraction or not. What is X-ray source? A few countries are lucky, and I will talk about the complete list, with their synchrotron facility where the X-ray source is much stronger. But if you look at other countries, rest of the world, the scientists have to depend on home sources which are not as powerful as the synchrotron sources. What is the problem? If you get a tiny crystal which could be diffracted and you could get data to solve it in a synchrotron source, you might not do that in the home sources. So, those challenges are also there. Making them together, at an average it takes 3 years for one protein structure to be solved. Also, with the new mutants, with one change in the nucleotide which is changing the amino acid, there are possibilities of change in conformation. Even a same protein could have been in a different conformation in its apostate. With the addition of different ligands, would have different interactions. So, you need to solve the structures of each and every candidates. That is the another challenge. Also, synchrotrons are very expensive, but if you talk about home sources, that is also costing 3 to 5 crore. All these added up to time and money. Coming to NMR, as I told, NMR have restriction in size. So, bigger proteins are not possible. You need very high concentration of the protein, and NMR for protein you need at least 600 megahertz. Megahertz is the power of the magnet. More powerful magnet provide you high resolution. Even if I consider a 600 megahertz machine to do all the settings, it would take at least more than 10 crore rupees. So, that is definitely a very challenging thing for a protein NMR specialist to convince the funding agencies or the institute to provide that much money. Also, as I told earlier, you have to produce your protein using multiple isotopes. So, that needs separate setup, that need you to pay much higher than normal production of protein. So, isotope labeling is another thing which makes it expensive. Coming to cryo electron microscopy, the first thing and which is very critical is the instrument. And when I am talking about individuals, you do not forget the initial cloning, expression and purification, that is applied to all the techniques. You need very pure protein to do all the things. Yes, in cryo you, as I already described, you go for complete cell or something like that, but when you are working on a protein, you need a purified protein. The instrument cost as an approximate setup 100 crores. Good that now more and more machines are coming in each and every country, reducing the price. So, I would say around 60 crores is the initial settlement money, and you could understand how difficult it for a cryo scientist to convince that the institute or the funding agency would invest 60 crore for one person or even a small group. So, those are the biggest challenges which makes the 3D structure determination difficult. Is there any other alternative? Yes, there is. There are structure prediction methods. Theoretical structure prediction, homology modeling, threading and ab initio. I will talk about them in details in the protein section and structure determination section. Now, a lot of talking is happening on AI based structure determination. Foldit have initiated, and now very recently AlphaFold have showing prediction of structures nearly at the level of experimental structures. So, yes, we will, it is not the course to discuss about them in details, but I will definitely introduce how and why they are working. And though people are thinking that this is the end of structural biology techniques, I will talk about what are the things where it could be utilized further and challenges further. I will finish this module with thing I talked in the first lecture of this module. There is a need of merger between basic sciences and biological sciences, and that initiative was coming when DNA research was going into advancement, and I invited all of you to talk about these marriages, and I have my own way to explain the marriage. In reality, I consider this merger as a marriage between basic science and biology. I consider this as a typical Indian marriage, because here two fields who are completely unknown to themselves coming together. They used to hate each other in normal situation. Basic science have their own ego, biological science have their own conventions. So, normal situation was not very friendly, and this is the first experience of live together with such an opposite entity. Definitely I am talking about a typical Indian marriage of my generation. Might be you guys are not agreeing with me, but that was in 90s, and a lot of people have gone through it and have very successful arranged marriages. So, the merger of biological science and basic science was going through the same phase. But you would say, why they come together if they have so many differences? Why they come together? Because it was an agreement on a common issue, as no other better alternative way could be possible to design. So, as they say, we got married because people would only give you gifts for getting married, not for getting divorced. Again, it's 90s concept, not like now when you see even the separation parties are happening. So, what I mean by that, biological science was the requirement, was always the need of next generation. You could have all the physical facilities, but it could not make any sense if you are not healthy. So, understanding biology, knowing about biology, doing research on biology, development of technologies to identify diseases, make people cured, was always the focus of the funding agencies. So, if you want to get fund, you have to come together. So, what are the problems for scientists coming from basic science background? If you look at biological problems are huge in comparison to others. It is very difficult to work on them without understanding them in detail. The systems are complex. There are loads of possibilities. Generally, when we say possibilities, they are binary, trinary and all, but if you look at biological problems, there are huge number of possibilities. It is not like yes or no, there are a lot of huge gray region in between. Percentage of confidence level is very low. My father is a doctor and he used to say that for a patient, when we say 100% there is a chance of cure, that means 66%. Think about 66% is 100% in medical science. So, when people come from basic science, they could not cope up initially with such divergence. A lot of biological phenomena are still mystery. We are doing biological research for such a long time, still we do not understand properly how a baby grows properly in the mother's womb. There are so many things, even with those trillions of dollars we are spending on cancer research, still we are unable to understand the cures. Hundreds and thousands of people are dying. So, it is really difficult to understand. And before I used to talk about antibiotic resistance in details, try to explain it to the people, but now after COVID, it becomes very easy. Even a common man understand what could be disasters with one tiny virus to swing its effect. The whole world is kind of closed down. So, I have taken the help of a cartoon where there is a conversation happening between Watson and Sherlock Holmes. Sherlock Holmes, with his typical way, is thinking, okay, biological science is elementary, but Watson, who is the originator, who is the person originating the function of Sherlock Holmes, he knows how difficult biological science is. So, he is saying, no shit Sherlock, it is not elementary. So, that is what is a problem to be defined. On the other hand, what are the problem for biologists? We should develop technological skills so that we could come closer to define the problems to people who are coming from basic science background. So, from the conversation it is apparently coming that a merger between biology and basic science is a near impossibility, but the distance they have, the problem could be solved if there is a introduction, and let us look at a solution. As I talked about typical Indian marriage, this is a photo from the marriage album. If you look at this photo and you know what it means by a Indian marriage, you will say that this photo is not complete, and you are right. The photo would be complete only when the bride and groom could be accompanied with the family. So, family in case of Indian arranged marriage solved a lot of problems, a lot of distances created, lot of misunderstanding would be solved by the involvement of family. Similarly, the merger of biology and basic science could be solved with the introduction of what we say interdisciplinary science. The introduction of interdisciplinary science plays revolutionary ways to solve the distances, to push them towards many path breaking innovations. So, let us take a look who are the scientists who came from different background and contributed to biology. I have already talked about people like Antony Fourcroy, Gerardus Johannes Mulder, Jacob Bacillus and Edward Buchner. They have contributed immensely to the protein science. There is Francis Crick, we have already talked about Watson-Crick model of DNA. Rosalind Franklin, she is considered as a tragic hero of science. She had really contributed immensely, but did not get her due. So, she is the first person to come up with the proper structural details of DNA. Erwin Chargaff, again I talked about ATGC, it is his contribution. And last in this list here, but not the least, definitely my favorite scientist Linus Pauling. Linus Pauling have so intense, so in-depth contribution in both the fields, in chemistry and biology. When I was growing up as a young kid, I was growing up as a chemist. I had the original version of his book about bonding theory and I was a fan. Then I shifted myself to biology research, and to my surprise I saw how tremendously he have contributed to again the field of biology. Talk about the protein structure, even talk about the DNA helicity, everywhere he have made immense contribution. I have talked about these guys already in details, but some of them we did not talk about, like Warren Weaver. He is a mathematician who coined the term molecular biology, biology at the molecular level, and he is the one who have called the basic scientists to contribute in this field, and he is the only mathematician to name a biological field. William Astbury, he is a physicist. He did a leading study of macromolecules by X-ray crystallography for studying structure of fibrous protein like keratin and collagen, and initially described the structural details of DNA in late 1930s, which have inspired Pauling, Watson, Crick to come up with the structure. So, Astbury have immense contribution. Maurice Wilkins, he was the mentor of Rosalind Franklin. and he is the one who took an early rough X-ray photograph in 1950. Raymond Gosling is a physicist. He is a student of Rosalind and worked on X-ray diffraction, took the famous photo 51, which is the X-ray diffraction pattern I have shown you earlier. He is the one who have actually take the photograph. Another two people, Jerry Donahue and John Gulland. Jerry Donahue is a theoretical chemist and John Gulland is a biochemist. They have provided the correct chemical information and advices to Watson and Crick for the DNA structure determination. Very excited to talk about Gopal Samudram Ramachandran, a physicist who have immense contribution. He is an Indian scientist, and Ramachandran plot, which I will talk in details, is one of the significant contribution towards the understanding of protein structure. How Ramachandran and his group have developed the science, the hypothesis of protein structure just based on the dihedral angles would really excite a young researcher towards higher science. And you would be even amazed when you see what are the basic instrumentations they have used. I would definitely talk about Gopal Samudram Ramachandran and Ramachandran plot in details. Also, we cannot ignore the discovering of triple helix structure of collagen by him. Another Indian scientist, Jagdish Chandra Bose, was a physicist, and he was the only physicist you could say who have contributed to the field of plant botanical science. He was pioneer in physics as well as plant science. He did the main research on microwave and radio wave refraction and made the first wireless radio signal detector. It was the primitive model of radio made by Guglielmo Marconi. He was also invented Crystallography, a device to study the growth and stimuli response in plants. With this, he have proved the first time that plant also respond and they have a life. Coming to John Haldane, he is a biostatistician mathematician who worked in the field of genetics and have a number of hypotheses. Stanley Miller, a chemist, Harold Urey, a physical chemist, they have done Urey-Miller experiment. These have shown us the origination of life, how life is originated could be explained by the Urey-Miller experiment. Samuel Banzer and Max Delbruck, who are physicists later turns into biophysicists, worked independently on first genetics and mutation. They both were inspired by the book What is Life, and if you don't remember, I talked about, I am going to talk in the next slide. What is Life is a book wrote by Schrödinger. So, Erwin Schrödinger, as I was talking about, a famous Nobel Prize winning physicist with many achievements in physics, wrote a book What is Life, in which he elucidated the biological and genetical concept from the physics point of view, and he was the early enthusiast of the field quantum biology. And when I talk about Schrödinger, I talk about quantum biology, I introduce Richard Feynman. You probably all of you know him. Like, what he talked about is a really significant prediction. What he told, certainly no subject or field is making more progress on so many fronts at the moment than biology, and if we are to name the most powerful assumption of all, which leads one on and on in an attempt to understand life, it is that all things are made of atoms, and that everything that living things do can be understood in terms of jigglings and wigglings of atoms. Coincidentally, Schrödinger and Feynman got Nobel Prize together. So, with that, I would finish the introductory module. In this module, I take an experimental approach. I have tried to link the history of biology with the advancement of DNA studies, how structural biology is contributed, and why or how the DNA research advancement leads to genome projects, genome sequencing, revolutionize the field of biology, but also produce a tremendous pressure on structural biology, help structural biology to grow in its way. In the next module, I will talk about the details of protein, how the protein sequence developed from amino acids, what is the chemistry, how the primary sequence of protein is converted to secondary structure, tertiary structure and quaternary structure, how it folds, who are the initial contributor to understand protein folding, how the protein folding is happening, what are the bonds, the non-covalent bonds we are talking about in details of that, how the protein folding is documented with a rule where Ramachandran and all are coming, how the protein structures are divided into domains, what are the importance of them, and overall how protein with its different structures try to play the role in different function. All of them could be discussed in the next module, proteins. With that, I thank all of you for listening. Thank you very much.